Sorry for the delay. Sorry for the delay. The computer wasn't seeing my computer. It was weird. Freaked me out for a minute. We're okay. Okay, quick business. Recitation starts this week. Some of you have already experienced that with me just about a half hour ago. Read the paper on Blackboard beforehand. Print out and bring discussion questions. You do not need to answer the discussion questions in advance. You can if you want, but you really don't need to because that's what you're going to be doing while you're there. Bring your Scantrons for your quiz and for your exams. I will collect all the exam Scantrons, and don't put your names on them because I'm going to redistribute them. Okay? Okay, so far we have been talking about what needs to happen or not happen to get a functional protein. We talked about transcription and RNA maturation and export from the nucleus. Then you have association with the ribosome and translation. We spoke a little bit about RNA stability, which we'll talk a little bit more today. And all this is to kind of understand the major concept of why are there more proteins than genes. For one gene, you might have multiple proteins resulting. And how can one protein have many different functions? That's the whole point. Because if you don't understand the concept of how you get to protein, then talking about like protein getting into the ER, going from place to place, doesn't make as much sense. Let me just open this. It's just seltzer water, just so you know. It'd be weird. Wow, forget it. Okay. I should get a cozy, right? We'll never know. What's Dr. Ehrlich drinking? So, I'm appropriate. It's just, I think it's got raspberry lime. Okay. So let's talk about, we're talking about one gene and they have multiple proteins. How does that happen? We touched on this, but we'll just talk a little bit more about this today. Alternative splicing. Who was able to tell me what alternative splicing was? Someone the other day raised their hand and said something about it. What's alternative splicing? We all know, alternative splicing can happen, but what is that? I keep forgetting to print out a class list so I can start calling out people. Yep.
Like the way in which the splicing chooses which exons are present.
Right, so splicing results in choice of which exons will be present in the resulting mRNA and then protein. So alternative splicing is there's some differences in which exons will be chosen, which splice sites will be recognized. Right. How do you think that happens? What do you think needs to happen to affect which exons remain, well not exons, really which introns are spliced out? What, like, if you had to come up with a mechanism, yes? Right, so yes, it's what the cell needs at the time that's going to determine what's going to be made. But okay, so the cell needs lots of protein X, and it's a result of alternative splicing. What do you think the mechanism, and I'm going to talk about mechanisms a lot, and what I want to know is this protein binds to this and does this. And you can say it in the most general sense, like this protein binds to this splice site and obscures it, or something to that effect, which is the answer. But yeah, so often what will happen when you need, so alternative splicing is not random. Well, I guess it could be sometimes, but ideally it's not random because no gene expression is. But often you'll have to, in induction of expression of some gene, you'll also maybe increase expression of a protein that might be involved in, for example, obscuring a certain splice site so that a larger exon is there in certain cases, which would be one way of alternative splicing being carried out. So the most extreme example is in Drosophila, where 38,000 proteins can come from one transcript. 38,000. I'm not talking about abundance, I'm talking about different proteins with different sequences. That's crazy, right? Who thinks that's insane? Oh, good, thank you. So there are different mechanisms. So one is splice site ambiguity. In that case, it's kind of random in the sense that splice sites are ambiguous, and sometimes some are more apt to be bound by the spliceosome, and sometimes other ones are more apt to be bound. And that happens more with genes that are constitutively expressed. What's constitutively expressed mean? I got you already today. Who else? Someone else. Constitutively expressed. Another word for constitutively is it's like your GRE. Yep. All in order, not all in order, all in a row. So if I say GAPDH is constitutively expressed, what does that mean? I heard someone shout out all the time. Right, so this would be a gene that's always expressed all the time. It's not highly induced under certain conditions. So genes that are constitutively expressed, meaning they're always being made, when you have alternative splicing involved in their production, it tends to be due to splice site ambiguity, meaning just sometimes certain splice sites are chosen and sometimes other splice sites are chosen, and there's kind of equal opportunity for the different transcripts to be resulting, and you get almost equal amounts of the different mRNAs. And then there can be this regulated splicing, which is the example I just gave, where you'll have a protein that its expression is upregulated, and its job is to bind to and either obscure a splice site, so it's ignored, or possibly recruit the splicing factors to a certain splice site. Those kind of things, so it's regulated. Okay. Yeah. Any questions about splicing, alternative splicing? The whole purpose of it is to get multiple proteins eventually from one gene. Okay, so another mechanism for getting many different proteins from one gene would be altered transcript cleavage and poly-A addition. So the 3' end of your mRNA is formed by the cleavage of the transcript. So you have RNA polymerase doing its thing, transcribing the RNA, and then at some point you'll have cleavage of the transcript. And based on where the cleavage happens, that can also affect what mRNA you end up with. So the example I have is in B cells. So B cells produce antibodies, right? Sure. Who's taking immunology? One. Okay. Okay, so B cells produce antibodies. So you guys know antibodies can be either membrane bound, so stuck to the top of the B cell, or secreted. And usually what happens is in a naive B cell, so a B cell that's never seen an antigen before, antigen would be like the bacteria or the virus or whatever's infecting you. Hopefully not self, that's bad. A naive B cell that's never seen antigen before is going to have membrane-bound antibody. Once it encounters antigen and it has to change and it becomes a mature B cell and it starts to secrete antibodies. And so the way this works is it upregulates the expression of a cleavage factor, and then it recognizes a poor cleavage site in the mRNA and cleaves the sequence that encodes the transmembrane domain. Very cool, right? So normally this will be, this is your DNA, and you've got these different exons, and then there's this poly-A site, and then there's this poly-A site. And so in an immature B cell, there's this membrane IgM, and this green part, these two exons, are going to encode for that transmembrane domain. And so normally it's like this, but after it becomes a mature B cell and it's an antibody secreting B cell, then expression of a cleavage factor will increase, and that will recognize this alternative cleavage site right over here, and will cleave off the transmembrane domain. And if you don't have a transmembrane domain and you're in the secretory pathway, you get secreted out of the cell. And so that's how that works. 2 proteins from 1 gene, right? Any questions? So just in case I was confusing, I have another way to look at it. Again, membrane-bound versus secreted antibody and B-cell maturation. This would be your gene, and you'd have one cleavage site for a short transcript, another cleavage site for the long transcript. And your long transcript is going to have your transmembrane domain, but a second cleavage site will be recognized in a mature B-cell, which will cleave off the transmembrane domain, and you get secretion of antibody. Do you need to memorize this? No, it's just a way to illustrate, to explain the concept of having different cleavage sites and how one gene can result in many different proteins. Okay? Questions? Okay. Another mechanism, this came up the other day when we were chatting, RNA editing. And I believe I had a question on the quiz about this. Insertion of U-nucleotides in RNA transcripts. So an example that's near and dear to me is ApoBec1, which is a cytidine deaminase. It deaminates, making cytidine uridine. And this actually changes the CAA codon to a UAA codon, which is a premature stop codon. And so when you introduce a stop codon earlier, is your protein bigger or smaller? Smaller, right? So you get a different protein. So here's your mRNA, and then your ApoBec1 is going to come and edit it and make your C to your U. And so in your long one, where you haven't had the editing, you get apolipoprotein B100, which is in LDL, as in low density lipoprotein. And then after ApoBec1 comes along and edits, it ends up being a completely different protein because you have this early stop codon generated, and then you have ApoB48, which is in chylomicron. And why would this need to happen? Well, at some point, under certain conditions, these different proteins would be needed. And what would happen is some kind of outside signal would say, hey, we need to increase the expression of ApoBec1 so it's present and able to edit this transcript because we need more of this ApoB48. Okay? So again, a mechanism for one gene resulting in 2 different proteins. Okay? Okay. So another way to regulate gene expression in a way, if you think of gene expression as being making mRNA and then resulting in a functional protein, is kind of regulating whether or not it can get out of the nucleus. So only 1 in 20 RNAs are exported from the nucleus, and the ones that are exported from the nucleus, they should not have any introns, and they should be capped and polyadenylated. So HIV, the virus, has a way of getting around this. It will actually, it has a protein called REV that will bind to a segment of the RNA sequence, because, so let me explain why this matters. You have to actually export the HIV genome, which happens to be RNA, from the nucleus, so it could be packaged to make virus, right? No, maybe. You want to make more virus. You're not a very successful virus if you don't end up making more virus. So, the virus comes in and it infects the cell and expresses its genes, but it also copies its own genome and makes virus parts to make progeny virus, which then come out of the cell and infect another cell, right? So it needs to export its own genome from the nucleus so that it can be packaged into new virus. But it's full of introns, and it's not capped or polyadenylated, so it shouldn't be able to make it out. And so it's evolved, viruses are smart, it evolved a way around it, and it has a protein called REV. And REV actually will bind to the RNA and interacts with the nuclear export receptor, which we will learn all about, and exports intron-containing RNA to the cytosol. Otherwise, there'd be no way to get the genome out to be packaged into the virus. So there are different fates of RNA, right? So we talked about all these different ways you can end up with different RNAs, which make different proteins. But then you can also think about, well, the RNA actually needs to get to the right place and associate with the ribosome in order to be translated and make a functional protein that's able to do what it needs to do. It's amazing we're alive when you consider all the things that need to happen just for proteins to get made. Right, so RNAs can be bound by ribosome and they can be translated into cytosol. Those of you who were in recitation today learned a little bit about how RNA containing a signal sequence is directed to the ER for the secretory pathway. Some RNAs will associate with cytoskeletal proteins that will move them to different locations so they can be where they need to be, where they're going to be used, where the resulting protein is going to be used. And that's determined by the 3' untranslated region, which is a sequence between the stop codon and the poly A. Can I repeat that? I didn't repeat anything. That? So I'm going to actually talk to you a little bit about it. Yep, so there's this region between the stop codon and poly A called the 3' untranslated region. It's untranslated because it's after the stop codon. It's 3' end. And that sequence can be recognized by different proteins and help to shuttle the RNAs to the places in the cell where it's going to be ultimately translated. So RNAs, they actually do localize to the parts of the cell where the resulting protein is going to be used. And so that's another place where things can go wrong in a way, right? If you don't get to the right place. So this is just right out of the book. They were looking, and I think it was Drosophila embryos, I believe. The 3' UTR determines localization of RNA. And when all they did was they took the RNA and they either left it normal or they deleted the 3' UTR. So the red, it went to a certain place in the embryo. And the green, you notice, is this diffuse staining. It didn't go anywhere. So that 3' UTR is important for localizing RNA to where it needs to be. So if you wanted to inhibit mRNA localization, what would you do? Right, you can delete your 3' UTR. So proteins can also bind to either end of the mRNA and negatively regulate translation. So it's another thing that can happen. All these different ways to control gene expression. Proteins can bind to the 5' or 3' ends, untranslated regions. So if you look at this, here's your AUG, that's your start, that's your stop. You can have proteins that bind 5' end or the 3' end, and they can negatively regulate translation. So that's also going to affect whether you get protein and how much of it you get. There are other ways also to affect translation. If you phosphorylate the translation initiation factor, EIF2, then you would inhibit ribosome assembly. If you don't assemble your ribosome, you can't translate, you don't get protein. Also, there are multiple AUGs that need to be recognized. If you have multiple AUGs, you need to recognize the right one. And then there are internal ribosomal entry sites, so places where the ribosome can enter and start translating. So now this is just kind of an overview of all the different ways that you can end up with different proteins, or whether or not you're even going to get protein from one gene. So now we've got mRNA that needs to be translated. But what if we don't even have the mRNA? We're definitely not going to get protein, right? So mRNA stability is another factor as to whether or not you're going to get protein. So most RNAs have a really short half-life, and they either degrade by just progressive poly A tail shortening, or a specific endonuclease might be upregulated that will cleave the poly A tail. if it needs to have a very short half-life. And then also sometimes you might need to increase the stability of certain RNAs. And there are ways to add to your poly A tail, so you're increasing your mRNA stability. So why might you want to play around with the stability of your RNA? And I don't mean you, like if you are a cell. Why might the cell want to play around with the stability of the RNA? Yeah. Damage, mutation. Well, think of it. Ideally, you don't even get to the point of making the RNA if things are that damaged. But think about it more in the process of, so all of what we're talking about now has to do with regulating how much protein you're going to get from gene expression. Yes?
If you make it more stable, you can make more protein from the same mRNA, or if you make it less stable, you can make less protein from a single mRNA.
Right, so if you have a very stable mRNA, then you can make more proteins. If some gene that needs to be expressed highly and for a long period of time, you would want that RNA to be more stable. If you don't want that much protein, you would make it less stable. So you made less protein from that RNA. I saw another hand up. Is there someone who wanted to add to that? Yeah. So I'm only half hearing you. So there's an external signal. When you make the RNA. Oh, so that's interesting. So there's an external signal that induces gene expression. You make an RNA, and then the signal goes away. What happens if you keep making that RNA even when the signal's gone? That's like unregulated gene expression, and you lose the regulation of gene expression. So it's actually important, once that signal's gone, for that RNA to degrade quickly. So you would want to maintain that half-life. But if it's some kind of gene that's expressed for long periods of time, then you would want to have to keep making more and more RNA. Okay, did I see another hand coming up? Okay. So then there's also nonsense-mediated decay, where premature in-frame stop codons can be recognized and mRNA is degraded. And then there is the degradation of RNAs that's controlled by these small RNAs, siRNAs, miRNAs, piRNAs. And they affect translation either through the direct degradation of RNA or the direct inhibition of translation. And they specifically target certain RNAs to degrade those specific RNAs. Yes?
Did you say nonsense or just stop codon in the middle of the RNA?
Yeah, so like a nonsense mutation. So if there's the premature in-frame stop codon, it'll be recognized and degraded. So with the siRNA pathway, it's just, this looks complicated, but the basics are just that you've got this POL2-driven transcription of a microRNA, which is then exported. It associates with some enzymes, and then you end up with this sequence that will base pair with the RNA that's targeted to be degraded. And you'll either get, with microRNAs, you'll either get translational repression or mRNA degradation. And with siRNA, you'll have these small double-stranded RNAs that associate with a complex that chops them up to like very small double-stranded RNAs, which also similarly will base pair with the target mRNA to be degraded. And these mechanisms are highly, highly used just in the lab. Like right now, as we speak in my lab, we're planning an experiment we're going to use shRNAs, which are pretty much these hairpins up here. You actually put in a plasmid that expresses this hairpin, which goes into this pathway to make these microRNAs to target a certain gene for degradation. Because our research question wants to determine whether or not a certain gene is involved in a certain process. And so the easiest way to do it is to experimentally express a microRNA, knock down expression of that gene in the cell, and then you can run your experiment in the absence of that gene. So this was another great Nobel Prize discovery. Who got the Nobel Prize for siRNA? Who's a big geek here who knows these things? I was never that person. There was always that person who knew who got the Nobel Prize for everything. I only know the people who've gotten them since I've been in grad school. Before that, I was like, what? Andy Fire. I think I tried to do no, that's in 409. I tried to do a paper with them. Okay, so that's microRNAs. Okay, so we talked about effects on RNA that result in different mRNAs. We talked about effects on RNA stability, whether or not your your RNA is long-lived or not. We talked about editing RNAs, all different ways to affect how much protein you're going to get or what kind of protein you're going to get. So now you have a protein, and there are ways to regulate whether that protein is active. Right? So you can have post-translational modifications. I only talked about that briefly when I was introducing the course probably 2 weeks ago. They affect protein stability, localization, interaction with other proteins. This is a list of different types of post-translational modifications. So I want you to memorize this for a quiz that's going to happen in about 10 minutes. Okay? Now this is really just to show you there's a lot of different post-translational modifications. When I say post-translational modification, what are we modifying? What biomolecule? Protein, right, thank you. Right, so you're modifying the protein covalently usually, right? Covalent modifications that affect stability, localization, interaction with other proteins, which all in turn affects the activity of proteins. These are important concepts to grasp. Yes? Does post-translational modification change the shape of the protein? In some cases, yes. Effectively, in some cases, yes. So if you think about it, let's say, what's the most common post-translational modification that you guys have heard about since 201? Phosphorylation. Right. So that's adding a phosphate group to either serine or threonine or pyrazine, a bunch of amino acids. If you think about it, if you normally, so let's make this go on for a minute. So I'm going to draw a protein, and let's say it looks like this, right? Because remember, there's these globular folded things that have a shape to them. And so let's say right here is normally where another protein likes to bind, right there, right? What if you phosphorylate right here? So now that protein, eraser. So let's say, so another protein likes to bind there, but let's say you phosphorylate something here. So you've got this big negative charge suddenly right there. Are you going to be able to still have this protein that likes to bind there? Probably not, if you understand the concepts of protein interaction, which has to do with fitting into a space and interacting with the right charges of the surrounding amino acids. So if normally there's this, positive, negative kind of charge that's allowing these two proteins to interact, and then you throw a phosphate group in there, there's something in the way, there's a change in the charge, it's going to probably inhibit that interaction. You could also have activating phosphorylation where it would do the same exact thing where maybe normally something wouldn't, Maybe something wouldn't normally bind there, but when you add the phosphate group, it makes this perfect binding site for, so there's your protein A and your protein B. So it's changing the shape in a way because if you think of proteins interacting with proteins, essentially it's like putting the pieces of a puzzle together. Does everyone follow that? Any questions? I took this class in grad school which was called something like protein structure and dynamics or something that I was like, I'm crazy for taking this, but I'm going to take it anyways because I had to get like X number of credits. And it was eye-opening in starting to really understand how protein structures work. And I don't know, I highly recommend if anyone's bored going and sitting in on the class just downtown at Hopkins. No one will notice, just hop in there. Okay, just don't take any exams. Horrible. Okay, so these are different post-translational modifications. And as we go through this course, I'll talk about, oh, this phosphorylation, and this binds to this, and this domain binds to this, and blah, 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 blah. And understanding this concept is really key to understanding a lot of that stuff. And you might be thinking to yourself, okay, so proteins are modified and that affects its binding stuff. Who cares? I don't understand. But you need to remember that proteins are these they're the machines of the cells, and they're doing jobs to help the cell function. And that usually is controlled by proteins interacting with other proteins. So if modifications are going to regulate whether proteins can interact or not, they're going to regulate whether processes can be turned on or off. Okay, does that make sense to people? I see some nodding. Is anyone does anyone think I'm speaking another language? I used to say Chinese, but then I realized I have some Chinese students, and they're like, oh, if you're speaking Chinese, I don't speak. Okay, everyone's cool? Okay, see me if you don't know what I'm talking about. Okay, so this is just an example of how post-translational modifications would affect the function of P53. Now we had someone raise their hand and talk about P53 the other week. P53 is this like guardian of the cell kind of gene that should be very low. When things go wrong, like DNA damage or all sorts of bad things happen, P53 tends to go up, which halts the cell cycle and causes apoptosis and all the things that are supposed to happen when the cell's in bad shape. But in cancer, P53 is highly mutated, so it doesn't do that, and then bad cells can survive. And P53 is highly regulated by post-translational modifications. And this is just showing you the million things that it does. So just in this figure, there's phosphorylation, which is the circle P's, acetylation, ubiquitination, sumylation, netylation. And there's all these different things going on with it. Sometimes P53 will be phosphorylated. Sometimes P53 will be acetylated. Sometimes P53 will interact with MGM2, and then it will be ubiquitinated. And then if it's ubiquitinated, and it'll be polyubiquitinated, they'll be degraded. And lots of stuff going on. That's one protein, and it's being modified in many ways and doing a gazillion different things. So a modification of one protein affects its function, or just regulates its function. Okay? In some cases, you'll have phosphorylation, which allows it to go into the nucleus. Sometimes you'll get sumylation to cover up sites for ubiquitination to self-stabilize. There's a lot going on. I just want you to appreciate how one protein can be regulated by post-translational modifications. I wanted to do a class all on ubiquitination. Would you guys take it? Okay, I'm going to ask you again at the end of the semester. Right before the final grades go in. No, so I keep telling people, I was like, I want to do an upper level class that's like all about ubiquitin. And people are like, why? And I'm like, well, it kind of controls everything, but no one realizes it until they have my class. And, well, maybe they should take my ubiquitin class. But you'll see. Okay, So right now you're going, what can I do with all this information? What is she doing to me? She's giving me so much information. Do I have to memorize all this? I can't write this fast. So I just want you to start categorizing things. As I give you lots of information, figure out a way to organize it. And usually you can categorize it based on some theme. So everything we just went through was pretty much processes that affect the production of a functional protein, right? And so then you can break that down into processes involving RNA, processes involving protein. And then to break that further down, processes involving number of mRNAs made, processes involving RNA stability, stuff like that. And you can start to group things and make some sense of it all. Any questions? I'll try to give you little tips about how to organize things whenever I think of it. I did a really good one last year with signaling, so someone will remind me when we go through signaling to help you with that. Because everyone was like, oh my god, thank you. It was fun. Okay, here's a practice problem. So you guys are going to stop for a minute and chat with your friends, but I'll go over it. So you are a scientist. It's slightly similar to something you thought about with this weekend's quiz. You are a scientist conducting HIV research. You notice that HIV infection results in lower levels of protein X, as seen by this Western blot. This is your marker. This is a time course of protein X. Here's protein X. Over time, there's less and less and less. Thick band, lots of protein, thin band, little protein. No band, no protein. What could be the reason for this? And then you might want to outline your thought process. So stop for a minute. I want you to take like 5 minutes and really think about what we just talked about. And yes, I know it was a lot, but someone somewhere got something out of it, hopefully. And come up with all the things that HIV might be doing to the cell that's resulting in these decreased amounts of protein X. And then we will discuss the class.
What part of the RNA so getting the RNA in the wrong spot. What about stimulating like mRNA? So let's say protein X is so there's always like a pathway for its insertion. And you untake the pathway. So in other words, you wouldn't even have, or you wouldn't have the mRNA being synthesized.
Okay. I would have given you more time, but people are going to just start to sit around. Who needs more time to discuss because you're so excited? Okay, so someone share an answer with me. There are multiple answers, yes.
I was thinking that maybe the HIV virus has some type of mechanism to remove the poly A tail so the mRNA making protein X is no longer stable.
Oh, I like that. That wasn't even one on my list. Very good. Yeah, so maybe the HIV protein expresses, the HIV virus expresses a gene that removes the poly A tail on this protein. That's really logical. What I want to say is, well, how would you do that? But what I'm thinking right now in response to that is, how would it target a specific protein? Because that almost seems very general, but I guess it's not general because the cell all the time specifically targets certain cancer. I'm just going through my inner conversation. Okay, yes.
The HIV virus could upregulate a protein that could bind to like the 3 or
So maybe it's up, okay, so it might be upregulating a protein that's binding to the 3' UTR, and what does that do?
would that inhibit the translation?
Inhibiting translation, okay, logical. Okay, give me more, that's another one that wasn't on my list. Good, you guys are thinking. Yep,
Having an affinity for a binding site on that particular protein that causes it to misfold and exposing the hydrophobic region that's supposed to be tucked in so then it goes into the proteasome and get degraded.
okay, so causes misfolding of the protein and induces degradation. Okay, and so is there another way to induce degradation other than causing misfolding? Oh, my children. Yes. Ubiquination, thank you. Yeah, actually HIV, so in my PhD, I joined the lab that was about to publish a paper in Science, like 5 minutes too late. Like probably Science magazine is like the best journal there is. It's like Harvard of journals, right? And so they were just about to publish this paper about how HIV has a protein that hijacks ubiquitin ligase and targets an antiviral protein for degradation. So that's exactly what the virus does with a few different proteins, and I believe we have a paper that we're going to do on that. What else? So we've got right now inhibit translation, trim off the poly A tail, induce degradation. What else? Could it be affecting splicing? Okay, so right now what you're seeing is a loss of protein. So who can give me a mechanism of how effective splicing would result in a decrease in protein? And you're not wrong, I'm just trying to get more detail out of you guys. Yeah.
Splicing earlier than you're supposed to.
Okay, so some sort of alternative splicing that doesn't make this protein anymore?
Now exons are missing.
Okay, so maybe, okay. Were you on the same page?
Yeah.
And what about you? Oh.
Can't you add something that if it's earlier to be
Okay, so you've touched on a few different things. Okay, so one is some kind of alternative splicing that results in not this protein being made. Because remember, splicing is RNA, is one gene resulting in multiple RNAs. So if you induce alternative splicing so that this RNA is not made anymore, yes, you'll have less protein. Sure, why not? You are talking about siRNA. So some viruses do encode their own microRNAs. Herpes viruses love to do this, which do target certain genes to be degraded, certain RNAs to be degraded, so sure. What else? Yep.
Could the HIV cause protein X to express the
Whoa. Could HIV increase P53? I mean, yeah, but you're going so far beyond one protein, then yeah, I guess. But actually, viruses, okay, so P53 causes cell death, so viruses are more apt to degrade P53 because viruses don't want to kill themselves. So, but I see where you were going. Okay, who else? I saw someone else over here. Yeah? No? Maybe?
I would say more about the RNA editing type of thing.
Right, someone said RNA editing back there too. RNA editing. Okay, right, so exactly if you, right, so if it induces RNA, some kind of RNA editing, and that results in not this protein being made, okay? Yep. Okay, so somehow affects the 3' ETR, somehow affects localization of the mRNA. Maybe, I guess then what you would need is for it to be degraded somehow, for the protein to go away. So that definitely could affect the function of a protein, but maybe not necessarily this. Yes?
Could it bind to the mRNA to prevent export from the nucleus?
Can it bind to the mRNA to prevent export from the nucleus? That kind of matches with the alternative splicing hypothesis. If you leave in an intron or something like that to prevent export, then it will be degraded, and that seems logical. There's still one very, very basic one. I guess I didn't talk too much about it, but I'm waiting to hear. Yeah?
If it somehow stops transcription.
That's what I was looking for. Prevent gene expression, stop transcription, right? Overexpress or repressor. Now, you guys, this is not 409, so we're not talking about regulation of gene expression. But those who have taken 409 or are taking 409, gene expression is regulated by the expression of transcription factors that will then recruit RNA polymerase to the DNA and activate gene expression. So if you either induce expression of an inhibitor or act as not an inhibitor, a repressor, or degrade an activator, something like that, that will also affect. So, this is actually a question I got asked during my oral exam, so I always like to give it to students because it's a good way for you to think through what you've learned and apply it to something, a real research question. Because this will come up, in my lab, these kind of things come up all the time where we'll put something and we'll be like, whoa, that protein just went away. Why did that happen? Let's figure that out. Because that's the kind of research I do. So, okay. Does anyone feel like they weren't heard? They had a really great idea? You got it all? Did most people follow that exercise and feel good about it? Okay. Okay. So now we have protein. Okay. Obligatory protein structure slide. I really don't need to even explain that to you guys, right? Protein needs to fold. Proteins are composed of amino acids. It's the chemical properties of amino acids that determine the protein structure. So you guys all understand that proteins fold based on their amino acid sequence. Those amino acids will have different charges or different properties. that are either going to allow them to bind to each other or repel or fold in certain ways, and that's how protein structure happens, right? These are your side chains. Who had to memorize all of these at some point? Okay, very good. I won't make you do it, but it is helpful if you work on this stuff. If you don't, then I'm sorry you have to do it. It's helpful. It's helpful. I actually worked in a lab where we had to make mutations, and without knowing your side chains, then you're kind of like shooting in the dark. So the biochemical properties, or the way I think of it most easily, is the different charges of amino acids determines the protein's secondary, tertiary, quaternary structure. And so you can imagine here's your polypeptide, and these side chains are hanging off, and you might have some hydrogen bonding here. You might have some hydrophobic interactions. Oh, no. You know, I just told myself, should I bring my... Okay, guys, I'm going to run to my office and grab my thing real quick. That happened during a job interview once. Okay. Hopefully we can plug it in somewhere. Please tell me there's somewhere to plug it in.
It's on the side.
Really?
Oh, no, no.
No, I wish. I wish. Oh, this is crazy in this day and age. No? A what? Plug anywhere plug? I could put it in here. Oh my God, guys. Ridiculous. You guys will get to relive this on the recorded lecture. Whew. So this happened when I was interviewing for jobs, and I'm pretty sure that's why I didn't get the job. But that's okay. It's okay, because I wouldn't be here at Towson if I had gotten that job. Okay. Well, maybe I would have. No, actually, no, I wouldn't have, because it was a totally different time. Okay. Disulfide bonds. Right. So we were talking about protein structure and amino acids having different properties, and it's these interactions between these side chains that keep a protein folded. And so, right, we had a disulfide bridge here, we have an ionic bond here. And so when you talk about proteins interacting with each other, it's through these kind of charge-based interactions. The easiest way to think about it. So proteins, they have to fold properly in order to function. That's why a misfolded protein is often sent to be degraded, because you don't want misfolded proteins hanging around too long, because they cause trouble. So you'll usually have your polypeptide chain, and then it will start to fold. Often you'll have these metal ions that will bind. I highlighted this because this week's recitation paper talks a lot about magnesium ions being important in the structure of the SRP. And I think that people mostly got that it was important with for the structure. Right, so usually you'll have it kind of folding the protein into a certain shape, at least in a certain region. You might get modifications that will affect its structure and its shape and allow it to bind to other subunits. Protein structure is defined by its sequence, so it's going to fold into a certain structure regardless, because it always wants to fold into a structure that has the lowest energy, where all your hydrophobic parts are on the inside. Nothing that takes a lot of energy to maintain the structure, right? So if you take a purified protein and then you treat it with urea, which is going to completely denature it, and then you remove the urea, it's going to fold right back into the same exact structure because it's based on the sequence, because it's based on those amino acids and their side chains. So protein folding, though, is it doesn't just spontaneously happen. Well, a small protein might spontaneously fold, but most proteins, as they come off the ribosome, so here's your RNA, here's your ribosome, here's your protein. Most proteins are bound by HSP70, which is a chaperone, which will bind to the protein and help to fold it. It'll help to stabilize the protein by not allowing all the different hydrophobic regions to aggregate and help it to fold. And then sometimes it won't just be HSP70, but other heat shock proteins or chaperones will bind and participate together to help fold the protein. And so one of these other ones is HSP60. HSP70 also interacts with HSP90 and HSP40 and all of these different heat shock proteins. Why are they called heat shock proteins? Who wants to guess? Yes?
Active at higher temperatures.
They're active at higher temperatures. Reverse that. Higher temperatures are, what do higher temperatures do to proteins? Denature them. And so that will activate the heat shock response. Cell goes, oh my God, we need to increase expression of all genes responsible for stabilizing our proteins and keeping them folded, such as chaperones. So they were identified as a response to heat shock, but that's not to, you shouldn't always assume that it has anything to do with heat. Like, for example, HSP90 tends to just bind to tons of different receptors and transcription factors and helps them to just remain stable until they bind to their binding partner. Okay. Things to consider when you're thinking about protein folding in vivo. It's crowded in there, right? It's not just like the pictures that you see in the textbooks where you've got your organelles and your one ribosome with your one protein. There's a lot going on in that cell, and so you really need these chaperones to help stabilize the protein while they're folding, otherwise they're going to just cause this big mess. This is an aggregate. They're just going to cause a big mess because all the hydrophobic regions are going to want to bind to each other. So that's why chaperones are important. You're having this simultaneous translation and folding. So you've got your protein coming off the ribosome, and it's folding as it's coming off the ribosome. And then there's high temperatures in the cell, and then there are also some degradation signals. So you really want, it's really important to have chaperones around to help proteins to fold. Otherwise, they'll aggregate, make a big mess, and be degraded, and that's not really good for anyone. So these are different chaperones. The big ones are HSP60, 90, 100, 70. All different chaperones. There are also chaperones that specifically work in the ER. We'll talk about them briefly. Chaperones that specifically work in the mitochondria, helping to bring proteins in. So there's HSP70. Again, binding to proteins as they're coming off the ribosome and helping them to fold. A correctly folded protein is good, but an incorrectly folded protein will likely be degraded. HSP 60, very different looking. It's almost the shape of a barrel where the protein goes inside and there's a lid or cap, and that keeps it inside and allows it to fold. Yep.
Is it really like, I guess the proteins, once they're completely, I guess, translated, the chaperones go on, or is it wild proteins?
It's co-translationally. As soon as so not with HSP 60, but with HSP 70, as the polypeptide is coming off the ribosome, HSP 70 is interacting. And that's what's helping to prevent this aggregation. Because if you think about it, until the whole protein's off, it can't really fold. So you need to stabilize it and so that the different domains can start to come together. Any other questions? Okay, so that's so they're both ATP dependent, HSP 60. So normally you'll have this folding, chaperone will get involved, try again, chaperone will get involved. If eventually it's not folded properly, you'll get degradation of that protein. So that's when there's no way to save it. So why would a protein be unable to be folded? Why would our genome encode a protein that was unable, like just never able to be folded? Well, it shouldn't, right? Mutations, right? So that's the big thing with mutations, well, sometimes, is they'll result in proteins that are misfolded and will get degraded. And I believe that's the big one with this cystic fibrosis, whatever receptor, CFTR. Okay. So we're talking about misfolded proteins. We may as well start with talking about ubiquitin then. But I just want to tell you that ubiquitin is not about targeting misfolded proteins to be degraded in the cellular trash can. I feel like I was always taught it that way until I went to college. I was like, oh, misfolded proteins, ubiquitinated and degraded. No. I mean, yes, that happens, but it's so much greater than that. Like I've said to you already, anytime a process needs to be turned off, usually there's targeted ubiquitination of a critical factor in that process, and that's what shuts off the process. So ubiquitin is a very small 76 amino acid protein. Some people call it ubiquitin tag sometimes because it's covalently attached to other proteins, so it tags them in some cases to be degraded, not always. It's covalently conjugated to other proteins. It's present in all eukaryotes, highly conserved. It's conjugated to lysine. So all the lysine residues have the potential to have ubiquitin added to it. And it has many functions, one of which is targeting proteins for degradation via the proteasome. Only one of which. So you're saying, what? Ubiquitin does more? Tell me about it. Well, I'll tell you. It's involved, so you can have ubiquitin in many different chains. There are people who study just the different chains of ubiquitin and what that results in. You can have monoubiquitination, which could result in endocytosis, affecting expression, and gene silencing. You can have these lysine 63 linked polyubiquitin chains that are involved in the DNA damage response. You can have the lysine 48 linked polyubiquitin chains. Those are the ones that target proteins to the proteasome. There's also the lysine 29 a little bit, but really when you're talking about proteasomal degradation, it's these lysine 48 linked chains. And you're like, well, what do you mean by lysine 48 linked chains? It's just where the ubiquitins are joined to each other. Right? If you have ubiquitin, and it's this protein, I made it linear. I know it's not linear. And so you'll have lysine 48 here, So that's gonna be where the next one joins in. And then you're listing 48, and that's gonna be where the next one joins in. Right, so that's ubiquitin 1, ubiquitin 2, ubiquitin 3, like that, and so on. That's what I mean by listing 48 linked. So each type of ubiquitination has its own meaning, it's kind of like its own code, and it tells the cell what needs to happen with the protein that's being ubiquitinated. Ubiquitin, I said it's a covalent, bless you, whoever that was. I said it's a covalent modification, so that means that there's usually enzymes involved with that. So there's the E1 enzyme, E2 and E3. E1, it just activates ubiquitin, ATP dependent. E2, that the ubiquitin is then transferred to the E2 enzyme, and then it's transferred to the E3 enzyme. The E3 enzyme is the most important one, because that's the one that recognizes the substrate that's going to be degraded. It's really important to know what you're degrading in the cell, right? Degrade the wrong thing, that's not good. There's no regulation there. So there's only one E1. That's pretty universal by ubiquitin activated. There's many E2s, well, a handful, but there's hundreds, probably more than hundreds, of E3s. And they all recognize specific targets and degrade those specific proteins only under specific circumstances. And so once a protein is ubiquitinated by the E3, ubiquitin ligase, then it's recognized by the proteasome, which will degrade it. And then there are deubiquitinating enzymes, which further complicate the whole thing because they can remove ubiquitin and stabilize the protein, or they can remove ubiquitin so it can be recycled, and then the protein's degraded. Any questions about ubiquitin? And so there's all these ubiquitin-like proteins. There's SUMO and ISG15 and NED, and they're all similar in that they have a similar structure, and they also are conjugated through this E1, E2, E3, and they're all post-translational modifications that affect the function of some whatever protein is being affected. Yeah.
So when you break those, it's all three of the responses that are degraded, or is it like E1, E2, and E3?
It's sequential. So ubiquitin is activated by E1, then it's transferred to E2, which transfers it to the E3. It's the E3 that recognizes the substrate. Then ubiquitin is transferred to the substrate, and then it's degraded. The substrate is degraded. Okay, any other questions before we say goodbye? Okay.